Welcome back to the channel. Today, we are diving into a fascinating study that explores the complex interplay between soybean plants and the soybean cyst nematode, a major agricultural pest. This research, titled The Soybean Rhg1 Locus for Resistance to the Soybean Cyst Nematode Heterodera glycines, regulates the expression of a large number of stress and defense-related genes in degenerating feeding cells, delves into the molecular mechanisms behind the Rhg1 locus, a key player in soybean resistance to this destructive nematode. The soybean cyst nematode, or SCN, is a serious threat to soybean production worldwide. This microscopic worm burrows into soybean roots and establishes feeding sites called syncytia. These syncytia, essentially modified plant cells, provide the nematode with nutrients and are crucial for its survival and reproduction. The economic impact of SCN is substantial, with annual losses in the United States alone estimated at over $1 billion. To combat the devastating effects of SCN, researchers have developed resistant soybean cultivars through traditional breeding. The Rhg1 locus has emerged as a crucial factor in this resistance. But how exactly does this locus work at the molecular level? This study sets out to unravel the intricate mechanisms behind Rhg1-mediated resistance. By understanding the genes regulated by Rhg1 during nematode infection, we can pave the way for more effective breeding strategies to develop soybean cultivars that are better equipped to withstand this persistent pest. To delve into the molecular mechanisms of Rhg1-mediated resistance, the researchers employed a rigorous experimental design. They utilized near-isogenic lines, NILs, of soybean, specifically NILR, which carries the Rhg1 locus and exhibits resistance to SCN, and NILS, which lacks the Rhg1 locus and is susceptible to the nematode. After infecting these lines with SCN, they collected root samples at 5 and 8 days post-infection for detailed analysis. To ensure accurate gene expression profiling, the researchers employed a technique called Laser Capture Microdissection, LCM. This technique allowed them to isolate syncytial cells with precision, eliminating contamination from surrounding tissues. The isolated RNA from the syncytial cells was then subjected to microarray analysis using the GeneChip soybean genome array, which includes probes for over 35,000 soybean transcripts. This analysis revealed a comprehensive picture of gene expression patterns in the resistant and susceptible lines. To validate the microarray results, the researchers employed quantitative PCR, qPCR, a highly sensitive technique that measures gene expression levels. Additionally, they conducted promotergus reporter experiments in transgenic soybean hairy roots to investigate the spatial expression of specific genes. The results of this study were quite revealing. The microarray analysis identified a staggering 1,447 differentially expressed genes between the resistant and susceptible lines. Among these, a significant number, 241 genes, were related to stress and defense responses. Notably, genes linked to stress responses, such as ATBAG6, a back domain protein involved in apoptosis, and several small heat shock proteins, HSPs, were significantly upregulated in the resistant line. These findings suggest that the resistant soybeans mount a robust defensive response to the nematode invasion. Interestingly, the syncytia in the resistant line began to degenerate by 8 days post-infection, while those in the susceptible line remained intact. This degeneration corresponds with the upregulation of stress-related genes, indicating that the resistant plants activate a defense mechanism leading to the collapse of the feeding cells. This observation highlights the dynamic interplay between the plant's defense mechanisms and the nematode's survival strategies. In their discussion, The authors emphasize the complex interplay between the plant's defense mechanisms and the nematode's survival strategies. While the nematode can penetrate the roots of resistant cultivars, its development is severely hindered due to the plant's induced defenses. This study highlights the intricate molecular network involved in resistance and underscores the need for further research to pinpoint specific genes within the Rhg1 locus and the downstream signaling pathways. In conclusion, this research provides valuable insights into the molecular basis of soybean resistance to SCN through the Rhg1 locus. By leveraging advanced techniques like LCM and microarray analysis, the authors have uncovered a network of stress and defense-related genes crucial for plant defense. This knowledge is essential for developing more effective breeding strategies to produce soybean cultivars that are resilient to this devastating pest, ensuring greater food security and sustainable agricultural practices. If you found this video helpful, be sure to give us a thumbs up and subscribe to our channel for more content like this. Also, if you are interested in creating engaging videos like this one, check out Collegites AI text-to-video technology. It's a powerful tool for transforming text into captivating video content. You can find the link in the description below.